Good afternoon, everyone. First, I would like to thank the section chair and the audience for joining this session. My name is Ina Ho, and I'm presenting our paper, Mining Electronic Health Records to Investigate Effectiveness of Ensemble Deep Clustering. Before we dive in the detail, I would like to express our gratitude to the U.S. National Science Foundation for supporting this work. Our work addresses a combination of clinical and technical challenges. On the clinical side, patient labeling of risk and diagnosis is inherently difficult. We heavily rely on diagnostic labels like ICD-10 code, but this kind of code have their own limitation and they are not always perfect. In fact, literature shows that medical misdiagnosis is the leading cause of death in America, meaning our clinical ground truth labels are often inaccurate or completely absent. At the same time, if we expecting a clinician to manually review a massive vector of lab values to decide on patient risk is incredible challenge. Let's move on to the technical problem side. For technical problems, when we look at current clinical research, the actual focus remains strictly depend on traditional k-means method. Meanwhile, even though there are some deep learning methods are proposed, but they are only proposed for image and text data. Most importantly, without a reliable ground truth, these cluster labels are hard to evaluate and then clinically interpret. We don't naturally know if cluster 0 and cluster 1 represents a high risk or low risk patient group. To build on this challenge, let's look at the current landscape of deep learning for tabular data, which has famously been called the last unconquered castle for deep neural networks. Recent literature consistently shows that tree-based models and traditional architecture frequently outperform deep neural networks on tabular dataset. Also, there is a recent literature survey on deep clustering. They mentioned that the reality remains strictly on traditional, largely relying on classical algorithms like k-means. While powerful deep clustering methods have been proposed recently, they have been developed almost exclusively for image, text, graph, and video data, but also they simply not as effective when translated to tabular health records. Therefore, we exploring deep clustering on tabular EHR data by proposing an ensemble approach for patient risk stratification in our study. Let's move on to the data we focus on in this study, electronic health records data. It contains an incredibly rich clinical history, capturing patient demographic, diagnostic code, lab test, and medication information. While standard machine learning applications in this domain are predominantly supervised using ICD-10 code, supervised models are fundamentally bottlenecked by data that is frequently incomplete, biased, or entirely unlabeled. So by leveraging deep clustering, we can map patients into a latent space, identify hidden patient cohorts, and stratify risk without introducing the overfitting or model bias that comes from supervised labels. Now let's see a review about how we extract and curate real-world EHR data. We get data from all of us research program, which is one of the largest biological data sources. It includes EHR domains, survey responses, and physical measurements. Let's see how we get the data. First, we select heart failure and patients who doesn't have cardiovascular diseases based on ICD-10 code. The second step, according to other heart failure related research and variable missing rate, we select 33 medical features out of more than 200 medical features from measurement table. And in the next step, we keep medical records whose date is closest to the disease's diagnosis date, and also we select same unit for each medical features, and we clean the outliers for the dataset. Finally, we get two cohort. Male groups has more than 7,000 patients with 33 features. Female dataset have around 15,000 patients. And for both of the dataset, their missing rate is lower than 1%. In this study, we benchmarked the different algorithm across three types. The first one is the traditional method. It has a k-means and the GMM. We apply it directly to the raw feature space. Second is hybrid method. An autoencoder is first trained, and then traditional clustering method is applied to that latent space. And third, we consider some deep clustering frameworks. We tested tested six image-based models along with G-cells, which is a unique model especially designed for tabular dataset. Since the traditional clustering model like k-means is still competitive, a notable weakness of deep clustering models, including G-cells, is the highly sensitive to the choice of embedding size. To overcome this concern, we introduced a novel ensemble solution. Instead of gambling on one single fixed dimension, we trained the independent G-cells model across a stepwise embedding from 2 to all the way to the full feature dimension of 33. We aggregate this diverse latent assignment using a mathematical averaging with the following equations for each sample G, creating the latent dimension ensemble. And take it for a step further, we combine this latent dimension ensemble with the raw power of traditional k-means and the GMM through a final majority voting schema. We call this approach the KGG ensemble. Let's review how deep learning model works and the evaluation method. We have our raw data X. Through an autoencoder, we get the embedding Z. We can get the X hat out of the decoder. Between X and X hat, we calculate the reconstruction loss. From the latent space Z, we apply MLP hat to get distribution Q, and another distribution P is generated from cluster head. And the cluster loss is calculated between P and Q. The real number of cluster of our dataset is 2. Between predict labels and true heart failure and non-cardiovascular diseases group, performance was evaluated using three metrics, which is accuracy, ARI score, and NIR score. Need to mention that to ensure completely fair evaluation, clinical variables are strictly hidden during training and tuning process. Now let's see the performance of our proposed method. The bold means the best performance, underline is the second, and the star means the third. First, let's see our proposed KGG ensemble method. The average rank of this model is achieves the best. And second, compare with the G-cells of a single latent dimension, G-cells latent dimension ensemble, average rank increases. Let's look at the traditional method, k-means and GMM. On the raw data, they proved to be competitive baseline, achieved the second and third best. However, notice when we apply the hybrid approach, running k-means on an autoencoder's reconstruction space causes a potential drop in the metrics. At last, let's see the image-based deep clustering method. Out-of-the-box image-based deep clustering methods performs poorly across the board. If you look at the dataset perspective, there is a large drop in the performance across almost all metrics when algorithms are forced to run on the combined male and female cohorts. This may happen because baseline clinical lab values and cardiovascular psychology differ fundamentally between biological sex. To visually validate this mathematical boundary, we project our high-dimensional space into 2D using t-SNE plot and a UMAP plot. For both visualization across all patient cohorts, k-means on the raw space shows a massive amount of overlap. In the contrast, G-cells yields more diffuse, cluster-friendly structures with visibly clear separation between patient groups. So to summarize the main findings of our benchmarking, first we demonstrate that standard k-means applied to raw data remains a remarkably strong baseline. Second, we highlighted a theory gap using a hybrid approach where you train the standard autoencoder for data reconstruction actually hurts cluster separation for traditional cluster models. Furthermore, we approved that out-of-the-box deep clustering methods fail on each records because they are inherently built for image, not for tabular data. Finally, our results scream the importance of a stratified analysis because clinical metrics were vary widely by biological sex, so splitting cohorts by sex is important for accurate clustering. This brings us to our conclusion and future work. We found that by spacing out the embedding dimension of G cells and averaging the assignment, we could decrease the model sensitivity to a hyperparameter choice. When we implement our Ensemble method, we have a state-of-art performance. KGG achieves the best accuracy, ARI, and NMI for both sex-specific cohort. So the overall takeaway is that the best clinical stratification is achieved when we consider biological sex difference and combine the complementary strengths of traditional and deep clustering method. So for the future work, we plan to use our Ensemble framework to discover subtypes of heart failure, and also we intend to validate the generalizability of KGG framework on other major diseases cohort. Here are the reference used in this presentation. That's all for my presentation. Thank you for listening. Please ask any questions.